Hi guys. We started. Did you guys get a chance to look over the test for this week? Yeah. We'll try to score now. That way we have a score every week when you do them. So please try to look it over ahead of the time. And we'll go over that. I'm trying to get these two copied out. This is the first question from your test. Tomorrow is holiday, right? You guys have a long weekend?
Yeah.
Wow. Some school gets 2 Mondays off. I think you guys get Friday and Monday or something. I think it's Washington's. I think it's Washington. Yeah. President's Day. All right. So let's see. I'm trying to get this so that is visible. Okay. All right. We'll get started, guys. So let's take a look at the question itself. Okay, so again, you're going to get 6 questions. These are older AP, so there's like 8, but you're going to get 6. Yeah. Okay. Hello, come in. So we're just going to go over the first test. Okay. All right. Okay, so the question here. So these are like spent over several pages. So the best way for these one on the test is that you probably want to just go back and forth and highlight the information. So here's a question. That year when this question showed up, it threw a lot of people off because AP Biology never covered much about, like circadian rhythm, an entire experiment on circadian rhythm. First thing is about circadian rhythm. This is basically the 24-hour cycles that our body goes through. One of the most prominent one is sleep-awake cycle. Right? You go sleep and you wake up in about 24-hour cycle period. Whether you're locked in a cave that has no light or not, you go through the same cycle. So this circadian rhythm is controlled by your hormones. Hormones that contain melatonin by your gland in the brain, somewhere in the middle of your head. But you don't have to know that hormone in order to do this experiment. So remember, don't be scared that if you see a question that you're not familiar with, you probably never heard before, but they'll find something that you can talk about that based on experiment. Okay, so you don't have to memorize. Circadian rhythm, you don't have to memorize the hormone. Melatonin is responsible for this, but this is important built-in mechanism that living things have. So this is also the reason why you have jet lag. So because the melatonin, the circadian rhythm is built in, but you can override that by having external stimuli like light. So for example, if you go to the Asia country and when it's supposed to be nighttime, you're supposed to be sleeping, but the outside daylight, okay, there's a bunch of outside daylight, so bright sunlight outside, and then that overrides the circadian rhythm response. So you gradually adjust to the new environment based on the environmental stimulus. So when you have circadian rhythm, going over the circadian rhythm, so you can see that circadian rhythm, the 24-hour cycle, you can also call that photoperiodism. You can also call that biological clock. They're very similar. Yeah. So this lab is about that. Okay. So we'll come and look at these in a sec. But what I want to put down here is that you might see the word circadian rhythm, biological clock, photoperiodism. They're all the same thing. Yeah. So circadian rhythm, RHYTH. Oops, THM. Okay. Rhythm. No, RY. Oh, hold on. How do you spell rhythm again? I forgot. What is it? RYTHE. No, RH. Yeah, RHYTHM, right? Oh yeah. Okay, yes, that looks right. Okay, so circadian rhythm or biological clock or photoperiodism. So you can kind of guess what these terms are, even though you're not taught specifically what they are. So, on the test, you might see things, words that you're not familiar with, but we have to deal with it as we answer the question. So these are the same thing. So this is controlled by hormone. So here's the thing about that. It tells us in the beginning, even though we're not sure, it says it's controlled by genetic and environmental conditions, including light. That is an important component here. So this would be something I would highlight. They usually do this. So if it's controlled entirely by genetic, that's it. Then whether you have light or not, it shouldn't matter your behavior, right? Your behavior will be identical. So the hamster's behavior will be identical whether you have light or not if it's purely genetic. But because of environmental conditions, like external stimuli play a role, so light can affect your circadian rhythm, just like having a jet lag, okay? So this is the genetic component. So researchers investigated effect of light on mouse behavior, not effect of darkness, but the effect of light, using a running wheel with a motion sensor to record activity on an actogram. So it's okay if you know what actogram is, but it's just activity. So you can see that what this means is that it's active on the right half and is inactive on the left half. And if you look at the figure to really understand, you have to read the figure to really understand what they're saying. So here it says strategy for recording mouse activity data. When the mouse is active on the running wheel, the activity is recorded as dark horizontal lines. And when the mouse is inactive, no dark line is recorded. So that's how you read it. So the first 6 hours you can see is there's no dark line, so the mouse is not active. And then 12 to 24 hours, it is active. So this would be because of circadian rhythm. Okay, all right. So here comes our experiment. So the next one here is okay, for the investigation. So here, adult male mice were individually housed in a cage in a soundproof room at 25 degrees Celsius. So here's the thing that caught my eye is the soundproof. It has to be soundproof in order to what? In order to minimize or eliminate actually by the effect of sound. So you cannot have sound or any other factors that comes to affect your experiment. When your experiment is only based on light and dark. So you're trying to test the effect of light. So darkness and light will be the only experimental factor. So this is how you eliminate other variables. Not only sound, can you give me another one, guys? What is another environmental factors that must be constant so that only the effect of light it is seen on the actogram? So there's sound, there's any environmental temperature, for example, right? Temperature, sound, any sources of food, right? Any other distraction. So you can just talk about those. Usually temperature, sound, amount of water, light, those are the big driving forces. Light, water, sound, pH, right? So pH doesn't really apply here. So smell, for example. So all these have to be constant. All right. So the mouse was provided with adequate food, water, bedding, running wheel. The mouse was exposed to a daily period of 12 hours. So you can see that's L, so 12 hours of light, and 12 hours of dark, so D. So L12, D12, for 14 days. So that's every day for 14 days. The activity is continuously monitored and is plotted here. So this is pretty good. See, this is exactly what we expect, right? So under 12 of light, there's no activity, and under 12 of darkness, there is. So this is like your control, yeah? An actogram of mouse activity, each row represents 24-hour period. So you can see from the very top is the first day, second day, third day, 12 hours of light, 12 hours of darkness. All right. So then after 14 days in L12, D12. So after the mouse has gone through this, so obviously is accustomed to this kind of condition, the mouse will place in a continuous darkness. So now there's no more light, which is all dark, BD, yeah? So the activity on the running wheel was recorded as before, and this is what the activity shows. Okay. So that's what we got. Okay, so now there's no more light. Remember our goal is to test the effect of light on circadian rhythm. So we have a control. After 14 days, accustomed to light and dark, now it's continuous darkness, no more light. And it seems like they're active here and there, right? Okay. So here's the actogram, okay, for that. So now we got to answer some of the question, yeah? So part A. So part A, the question for part A actually has nothing to do with the experiment. It's just a biological concept question. So the nervous system plays a role in coordinating observed activity pattern of mice in a response to light-dark stimuli. Describe one role of each of the following anatomical structures in responding to light-dark stimuli. So the one you talk about photoreceptors in the retina. Retina is the back of the eye that has cones and rods. Okay, that's retina. Okay, that's how we see. Cat's retina has a mirror-like reflective ability so that they can see in the dark. So that's why they glare at nighttime. So retina is the back of your eye. Okay, so if you ever been to the optometrist, they'll look into the retina. You can see the entire retina to see the blood vessels. Are they okay? If there's any potential medical issues. All right, then the brain, right? Brain is the central nervous system that ultimately controls everything, and a motor neuron. Okay, so like that. So you got to describe one role. So just not state one role. But if I were to state one role, the brain would be integration. But to describe one of the roles, then you want to say that the brain receives information about light from the sensory neurons, and then integrate and process and make a decision what to do. That would be a description, right? But a statement would be just integration of sensory input, right? So I guess, yeah, so it just it's a fine line between describing and statement. So a little bit longer than just stating the answer. So that's what you have to do here. So photoreceptor in the retina receive information, right? That's a reception. And you see the brain is the integration and the motor neuron is the output, right? So you always have that three things. You receive information using sensory receptor, the protein molecules, they're able to transduce external stimuli into a nerve impulse. Nerve impulse will be sent in by sensory neuron to the spine first, to the brain. Fruit fly does not have spine, but mammals like mouse do have spine, right? And the brain will receive that information using the proper processing centers, integrate that information to make the best adaptive decision, right? And the motor neuron will carry out the command from the brain, right, so that the mouse knows what to do. Either it is active or it is not. So you'll put something like that together. Is that okay? Yeah. So the key part here is, guys, you have to learn how to put that in words. So even after I just mentioned, I just talked about it, you have to be able to put that in words. Because sometimes when you think of a concept in your head, it is easy. But once you start typing it out, it becomes more difficult. Yeah, it happens to me as well. It happens to me because if I were to answer that question now, okay, so this is basically the result that we saw. Okay, so if I were to type that question right now, probably won't remember what I said. Then I have to kind of like retype it out. So you have to learn to do that. So this is why we take tests so early. And it feels like I'm not ready to take a test, but I think you guys are. These are things that is common sense. You don't need to read the entire nervous system or endocrine system to answer the first part, right? Okay. So usually I would, so I was thinking that if you guys want, what you guys should do is that if you can bring your laptop here to type it out, or you can use scratch paper, but the key is to be able to put that in words, okay? So again, you want to describe the role of photoreceptor. So anybody want to try? How do you describe the role of a photoreceptor, right? This. Okay. So again, you can start by putting some ideas like outlines, but remember you're rushed for time on the test. So what is the role of the photoreceptors to receive stimulus, right? Light. So if they use light, talk about light specifically. External stimulus will be too general, right? Light. Okay. So we have the input, and then we have the output. Sorry, we have input and integration, and then output. That's basically what the question is. Yeah. Integration and output. So, so what is a role? Photoreceptor. Photoreceptor. Okay. Senses. Okay. Detect and senses light or dark or absence of light, and transform or transduce, right? So signal transduction, right? Transduce the stimulus into nerve impulse or nerve impulse, action potential, if you want, right? Via sensory neurons, okay? And send information to its brain. The brain is a central nervous system, okay, that integrates the external stimuli like sensory input, yeah, if you want, okay. So so if you haven't done it, make sure you are practicing doing this, yeah. So our goal is to make sure that you're able to write. Okay. So the brain is the central nervous system integrate sensory information or sensory input. Okay. So light and dark information, okay, and come up with the best decision on what to do, right? So I'm using more general words instead of saying that you know, the most adaptive solution to the sensory information, right? So basically, they come up with the best solution decision on what the mouse should do, okay? On what the mouse should do, right? So should it run? Because there's another response. If you were to flip over a rock, a lot of the bugs runs around. That is called Texas, yeah. So a lot of the they slide, try to hide dark place. This is an adaptive response because usually that happens when something is flipped over, light comes, you turn on the light, usually when danger is around the corner. Okay. So the next one, motor neuron. Motor neuron, okay. Motor neuron, so I'm just gonna say carries out, okay, command by the brain. And then, so we know that's what motor neuron does, but then talk about the muscles, right? The effectors. And the muscles and sends carry out command by the brain and send it to muscles of the mouth. So that would be okay. Just a little bit description, yeah. So don't stress too much. If this is something you feel pretty easy, then put something out and then move on. So you can see that when I type it out, it's a little bit different from what I've said. Yeah. So take a little bit of time to kind of get used to. Okay. All right. So again, sometimes I'm not gonna type out all the answers. So please, I asked the office to put the video link. So if you want to review the test, just kind of look over the videos, you know, fast forward to the part that I'm talking about the question. So it'll be easier, yeah? Okay, but this practice is important, okay? So part B. So based on the analysis of the data, describe the activity pattern of mice during light and dark period of the L12, D12 cycle. So make sure you go to the right cycle, yeah? Because this one is in the L12, D12, not the complete darkness, DD cycle. So describe the activity pattern. So that's super easy because the mouse is active. So here's what you cannot do. This is the one that you want to describe, right? This is the L12, D12. So it said describe the activity pattern. So you can say that the mouth is active in the dark and inactive during the light. That would be suffice, yeah. But if you want to be a little bit more detailed, you can say that the mice is inactive during the 12 hours of light, right? And becomes active during the second 12 hours of the day, right? So during in the dark. So mice is inactive and active. That's it. So that's what this is telling you. That little dots there, don't worry about those. Those are outliers, like statistically insignificant compared to the rest of the data. So those activities, all of a sudden, maybe the mouse was twitching, dreaming or something, right? We do that too. So, and it just woke up and start running on the wheels. Okay, so this is part B. Mouse. Okay, so again, I'm gonna try to put it up. Okay, so mouse. If I spell something wrong, okay. Okay, so mouse is inactive for 12 hours and becomes active. Okay. Yeah, this is the thing I don't like about this is that I can becomes active. They have to make this a much better function. Okay, I can't move it up. I have to. Oh yeah. Okay. So it becomes active during the second half. 12 hours. That's it. Okay. So super easy. Part C. You see that you don't need to know the entire nervous system at all to do this. Okay. It's crazy. You just have some common sense and some understanding of science. All right. Part C. Let's see what part C wants. So it gets a little bit harder. So part C, the researcher. So again, one of those claims. Either the researcher made a claim, you got to support it, or some student made a claim, you got to support it. So the researcher claimed that genetically controlled circadian rhythm in mice does not follow 24-hour cycle. It described one difference between the daily pattern of the activity under L12D12 and under double D. Okay, so describe one difference between those two charts and use that data to support the researcher's claim. Okay, so why don't we describe one difference first? What is the difference between what we saw? Okay, like right here. So between this one, which is in double D, and then this one here, right? So obviously is the pattern, right? The L12D12 has a distinctive pattern, right? The mice was inactive during the day when there's light and is active at night, okay? And that's what mice do. But this one in continuous darkness, it doesn't seem, you just want one difference. So there's no such pattern. It seems like the mouth is active without pattern, right? Okay, you don't have to try to describe the pattern. Sometimes it's too hard. The first thought is that, should I describe this pattern? Because it seems like there may be a pattern. Yeah, there may be a pattern. They're active earlier and earlier during the day. Remember, 0 is on the left side, right? So the first day they're active about 8 hours. And then the second day they're about 7 hours. And then become earlier and earlier throughout the day. So do you have to use that pattern? Yeah, that's fine. That's one difference. Or you can just say there's not really much pattern. Okay, so that's it. That's just to end that point. Yeah. So one of the differences that, let me put it here. So mice activity, mice become active. Yeah. Mouse becomes active earlier and earlier in the DD condition. Yeah. So they call condition. I'm trying to use whatever the word they gave you. Yeah. So under the DD conditions, then the L12 D12 condition. Yeah. So you can use some of the wording they use. Yeah. And then if you say earlier, you gotta put then what? Earlier than what? Otherwise it's ambiguous. Yeah. So it's kind of obvious, but you know, you have to say that. Okay. So that will be one difference. Okay. All right. And so let's look at the next one is we got to answer this more, right? So the claim is that genetically controlled circadian rhythm does not follow. So how do we support the claim? Now, if genetic control, if circadian rhythm does follow 24-hour cycle, then when we switch off all the lights, shouldn't we get the same pattern, right? Because it's genetically controlled, not by light, not by external stimuli. So if indeed circadian rhythm is controlled by genes, that means something you cannot change. You're born with it. So wouldn't it not matter whether you're in complete darkness or light and dark? It shouldn't matter, right? Okay, does that make sense? So use the data to support the claim. So the data, so you just say that's basically what we said, right? Okay, so the research is correct. Yeah, it does not follow 24-hour cycle. If it does follow a 24-hour cycle, we should get similar data as the L12D12 in the double D condition. So if you look at the scoring guide, guys, so always good to look at the scoring guide as a review. So you can see that this part here, we talk about part C, right? So what is the support, right? And also the description. See that the description for the double D, you can say active period begins a little earlier, sure. And it's shorter than 12 hours. So yeah, that's another pattern. It was a little bit shorter than 12 hours. So, and then you can even say the last bullet, I like that one the best. Pattern of activity shifts each day. So they become active during different times of the day. Okay, and then here's the support for the researcher, yeah? So if you look at the second bullet, if it were a 24-hour circadian rhythm, the pattern DD in DD would be the same, right, as activity pattern in L12, D12. Again, the point of genetic control means that you're born with it, there's nothing you can change regardless what the external stimulus is, light or dark, doesn't matter. So you should behave the same way, okay? You should behave the same way. Whether you're old, whether you're young mouse, whether you're sick, whether there's light or not, you always have the same pattern because it's genetically controlled. You cannot change your gene. So that will be the answer, yeah? Okay, so let's just move on. So you can see that you can tell, AP Bio is very easy to tell how many points. So part A, every bullet is 1 point, right? Part B, describe is 1 point. And part C here, you have 2 points here, right? Describe and then support. Right, let's look at the next one. Part D. Part D, to investigate the claim that exposure to light overrides the genetic controlled circadian rhythm. So that's like jet lag. The researchers plan to repeat the experiment with mutant mice lacking a gene that controls circadian rhythm. So now you have an experiment with mice that don't have the gene. So these mice have no gene. Okay, so we're going to compare, predict the observed activity pattern of the mutant mice under O12, D12 and under double D conditions that will support the claim that light does override. Okay? So we're saying that light override genes, which seems to be the case when we have jet lag. Ultimately, we are accustomed to the circadian rhythm in the new environment. Yeah. So how would a mutant mice, right? Now, the mutant mice does not have this, so there's a lot of possible answers. Yeah. So if you look at the scoring guide here, okay, for this one, there's a lot of possible answers here. So you can see that here. So let's look at the first row. You see the first row is mutant under L12, D12. So that means light and dark. See, it says the predicted activity will be normal rhythm, similar to the wild type under LT12. Why is that, guys? Why would the mutant mice, again, the mutant mice means the mice does not have the genes to control circadian rhythm. Why would it be, your prediction would be normal, right? That will be identical to or similar to the wild type. Well, the reason is that if you are not genetically controlled, then you must be controlled by external stimulus. That's the presumption. Because we're told that circadian rhythm is controlled by both the genes or by the light and night cycle. So if you don't have the genes because you're mutant, to control circadian rhythm on your own, then your circadian rhythm is entirely controlled by light or dark. So in the light, you are not going to have activity. In the dark, you will, because you have no ability, no genes, right, to control circadian rhythm on your own. Okay, so this is the best prediction, yeah, and actually, this is the only prediction, yeah, that you can do for L12, B12. Once again, circadian rhythm, waking up about 24 hours, is controlled by both light and dark, and also by the genetics. If you get rid of the genetic component, then the only thing that's left is light and dark. So this mutant mice without the genetic component would have to be active during the night and active during the day. Yeah, you don't have to say it's similar to the wild type, but you can just say it's active during the night and inactive during the day. Now, what about this mutant under DD? So then the mouse is lost, right? So it doesn't have the genetic component to control circadian rhythm, and there's no more light to signal inactivity, right? So then you can just say whatever. So the mice would either have random activity throughout the 24-hour cycle. There's no much pattern, either one of those bullets, right? Or the mouse is now constantly active, because nighttime is usually when they're active. They don't know when to stop. So there's no right or wrong answer for this one, okay? Because the best guess is that there's not much pattern. You can also say that they become active, more active than when they're not. When they really exhaust themselves, they become inactive. So they're constantly active until exhaustion or something like that. Okay, okay. So that's the genetic aspect. So, so the clue for this question, once again, is from maybe the first part, right? It says that the circadian rhythm is controlled. So if you look at the this sentence here, circadian rhythm controlled by both genetic and environmental conditions, kind of like what we know, right? Okay, so that will be part B. Okay, let's look at the next one. Yeah. So the next one here is part E, last one. So in nature, mice are potential prey for some predatory birds like owls, right? That hunt during the day. So for owls hunt at night. So other birds then, they come and eat the mouse, hunt them during the day. Describe two features of a model that represents how predator-prey relationship between bird and the mice may have resulted in the evolution of the observed activity pattern mice. So the one have two features. So you can describe two features. Talk about two things that you know. What two things? The predator-prey relationship, yeah, and then in terms of evolution, right? So why do mice basically active at nighttime and inactive during the day? Yeah, so you want to describe two things about predator-prey relationship. That's like ecology, that and also the evolution of this. Why does mice behave this way today? Why are they active at night, inactive during the day? So sometimes that can give you a little bit clue. There's questions in there. If you're mice, you're more adaptive if you are active at night and not active during the day when the predatory bird that hunt during the days are around. So this has to do with adaptation, survival, right? Okay. So definitely that would be something. So this is two features, so you're gonna have 2 points. So if you look at the answer key, okay, so here, so the answer, and I'll also post the answer key, yeah. So describe two features. So the two features is talking about selective advantage and reproductive success. So that's pretty good. Yeah, I like those two words, but you got to describe it because the question says to describe. So what I mentioned was selective advantage. That there's a positive selection for mice that is active during the night when there's no predatory birds, right? So that's what the first bullet is saying. Okay, there's definitely a selective advantage. Yeah, or you can say there's a selection against mice that's active during the day when the predators are around. So they'll be selected against. So either way, right? So you can talk about this, right? Then what about the second feature? So it's hard to come up with a second feature. They smartly have done that, talk about reproductive success, because that's That's also what evolution. So you got to think about Darwin. Darwin talked about what did Darwin talk about, right? Darwin talked about there's variations. Okay, so that doesn't apply here. There's tons of variations. So there's always some mice that is active during the day, some are active during the night. There's tons of variation. There's overproduction that doesn't apply here, and there's limits to resources, doesn't really apply here. And then there's natural selection, right? So the most adaptive mice, which is the selective advantage. And then there's differential reproduction. So the mice that are selected for survive to reproduce and leave more offspring with the same trait. And that's why the present day mice have this kind of trait that they are active at night only. Because all the ones that are not active at the night and active during the day are eaten by the predatory bird. So they don't survive to pass on that trait, right? So this would be the next level thinking beyond just natural selection. So they force you to think about natural selection. So you just regurgitate whatever Darwin said and to see which point may be used as a reasoning here. So even if you didn't get the second feature, you can probably get the first one, which is the easiest one, to avoid predation. That's why they're active at night, right? Okay. So any one of those bullet points, and this is two features, so there's 2 points for each. So if you got stuck on the second part, couldn't come up with anything, just write something and then come back to leave enough space in case that you don't have time to come back. You can write something that you think is the best answer and then modify it later when you do have time. Yeah. All right. So there's that. This is about 10 points. Yeah. Okay. Let's look at the second question. So remember, the first two are the biggest ones, and the biggest ones is the one that you want to answer correctly and put more thoughts into. If you get stuck on the biggest one, move on. Okay. Don't waste like 20, 30 minutes on one question. You're gonna run out of time. Go get to the easiest one towards the end. So this one here, if you look at the question, this is the data that's given. So figure 1 is glycolysis. So we're talking about glycolysis and oxidation of pyruvate. Figure 2 is Krebs cycle in the matrix of mitochondria. And figure 3 naturally is electron transport chain. Yeah. So you don't have to memorize electron transport chain. You're given that, right? Remember the electron donor. One of them is NADH. Okay? Donates electron. Electron gets transported from complex 1 to 2 to 3 to 4, then to oxygen. Oxygen is the final electron acceptor. And you use the energy stored in the electrons to pump hydrogen ions across the inner membrane of mitochondria to create a proton gradient so that chemiosmosis can occur. And you can see the chemiosmosis is actually not drawn out here. If you were to draw chemiosmosis, you probably have to extend the membrane. And this is chemiosmosis right here, right? You have this ATP synthase. The protons will flow back, kind of like water flows down waterfall, releases energy that can be used to synthesize ATP from adenine diphosphate and a phosphate. So this is how ATPs are made. This is called chemiosmosis. Okay, so they decide not to draw that in for you. And you don't need to memorize what is produced by Krebs cycle. So if you look at the diagram, what exactly is produced by Krebs cycle in terms of energy? Well, you can see that acetyl-CoA goes in, you produce 2 NADH here, 2 NADH here, and 2 NADH here. So that would be a total of 6 NADH. You produce 2 FADH2. Those have energy, right? And then you produce 2 GTP, which will convert into ATP later. So that would be definitely ATP by substrate level phosphorylation, right? And you produce 2 waste gases, CO2 here, and then 2 more, a total of 4 carbon dioxide. Do you have to memorize it? No, this is not about memorization, right? And also glycolysis, you can see glucose become pyruvate. There's 10 steps here, but they don't want you to memorize all the steps. They just want you to know glycolysis net 2 ATP and netted 2 NADH. And when pyruvate converts to acetyl-CoA, you get 2 more NADH and 2 carbon dioxide gas released. So you can see for 1 glucose, you release 2 CO2 here, 2 more, so a total of 6 carbon dioxide. And that's what this overall equation for cellular respiration tells you, right? If you were to balance the cellular respiration, you're going to get 6 CO2 because of this. And you're going to get 6 water, it's not shown here, and you're going to require 6 oxygen molecule. Okay, that's a very quick review for cell respiration, but that's given. Now let's see what they want here. So number 2, cell respiration includes metabolic pathway of glycolysis, cycle, and electron transport chain as represented. In cell respiration, carbohydrate and other metabolites are oxidized. Remember, oxidized means extracting electrons from glucose, and that's how we were able to get energy, right? So we oxidize glucose, 4 glucose electrons, put them onto NADH, FADH2s, and then we run electron transport chain to extract those energy. So the resulting energy transfer reactions support the synthesis of ATP. Sure, that's what we're seeing here. Okay, so the question, number 8. Okay, let's take a look at that. So I'm gonna make some room here. So again, I'm not typing out everything, so that you know. All right, so let's look at question number 2. So let me take a picture of this. So it's difficult to take picture when the screen is kind of small. Yeah, see, I can't even find the icons. Okay. Where's my icons? So stupid. Oh, there we go. Okay. Needs to be out. And it's blocking. Okay. I'm not gonna take a picture. We'll just do this. Okay, move it to the side. Yikes. Okay, so let's look at this one. So use the information. So here, what we need to do is that describe one contribution of each of the following, yeah, in ATP synthesis. So again, ATP synthesis is the goal. What is one contribution of each of the following? So if you look at the first bullet, so what is the contribution of the catabolism, means to break down, catalyze catalysis, hydrolysis, right? So break down glucose in glycolysis and pyruvate oxidation. So how does that contribute to ATP synthesis? So it seems like this question is kind of obvious. Glycolysis produces 2 ATP directly. That's the contribution. You break down glucose directly by substrate level phosphorylation. I will use that word if you remember it. If not, it's okay. Just tell them that glucose, the oxidation of glucose and pyruvate oxidation produce 2 ATP directly. It directly contribute to ATP synthesis, right? Without going through electron transport chain, without going through all that difficult aerobic respiration, yeah? Because glycolysis is able to produce ATP anaerobically, yeah? It does not require oxygen to produce ATP. This is why glycolysis is the most ancient way of energy production, yeah? Okay, so it's the most ancient method of ATP production, okay? Alrighty, so this is giving me a lot of trouble, okay, just to get to these. So that's basically it. And then if you look at the oxidation of intermediates in Krebs cycle, kind of similar, right? So again, you just read the diagram. So oxidation of intermediates in the Krebs cycle, how does that contribute to ATP? It's the same thing, you know? When Krebs cycle is oxidized, all these intermediates, as they go through the Krebs cycle, you produce 2 ATP equivalents, which is the form of GTP, yeah? So directly, also by substrate level phosphorylation, yeah? So both processes produce ATP directly without using electron transfer chain, okay? So that's basically the reason, yeah? So let me see, let me just put some random thing here. It's so frustrating, this thing. It's so slow, and then I can't get to this. Okay, let me just put that like this. Okay. And the last one must be talking about the electron transfer chain, yeah? So the last one here, metabolism. Okay. Glycolysis. So you can even combine both questions together, yeah? Makes ATP directly via substrate level phosphorylation, okay, directly using electron transport chain, yeah, okay. Without using electron transport chain, okay, or chemiosmosis. You don't even need to mention those words, yeah, I'm just not, okay. So the second part though, so I actually combine both of them together. You shouldn't, you should separate them, yeah. If you combine both of them together, it's fine too. I've seen a student do that, okay, as long as you address them, right? So oxidation of intermediates releases the energy. So I'm actually going to separate them. Glycolysis makes ATP, right? Okay. Then second one, oxidation. Intermediates releases. I'm going to say releases energy to make NADH and FADH2. So that could contribute, right? Which ultimately results in ATP production, which lead to ATP production. That will be fun too. So I don't have to focus on the GTP. Yeah, I can focus on NADH, FADH2s. Ultimately, will be turned into ATP. Right. Okay. The last one, formation of proton gradient by electron transport chain. Yeah, the proton gradient, right, is a form of potential energy. Yeah. ATP synthesis, ATP synthase, used to synthesize ATP. Chemiosmosis, right? If you know the word, use that word. Yeah. There's a lot of ways to do this. Yeah. It's kind of silly. Osmosis. Okay. So let's look at the scoring guide. If you look at the scoring guide here, it's just you write something, you get a point. When they ask you a question like, why is this obvious? Why are they asking obvious one? They just want you to be able to put into words. So let's look at all these possible bullets. The first one, catabolism of glucose in glycolysis. You can say it produces ATP for ETC. You can say produce acetyl-CoA for entry into the Krebs cycle because only acetyl-CoA can enter, right? And you can say provides energy by substrate level phosphorylation, by phosphorylating ADP. And intermediate, same thing, produce NADH, FADH2 for electron transport chain harvest, right? We can talk about anything, yeah. And the last one is the flux of ions generate ATP by chemiosmosis. All right, that's it. So super easy. 3 easy points. Yeah. Now, part B. Each of the following observations. Use each of the following observations. So they give you 3 observations to justify the claim that glycolysis first occurred in a common ancestor of all living things. So this is important. Every time you want to justify a claim, sure, yeah, you understand what the claim is, right? Okay, so what is the claim here? Yeah, justify the claim that glycolysis first occurred in a common ancestor. So that means glycolysis is universal. That means glycolysis is in every single descendant, right? So if you look at the first bullet, yeah, nearly all existing organisms perform glycolysis. So use this to justify it. Under anaerobic conditions, and glycolysis occur only in the cytosol. Okay, so because the observations are so similar to your justification, you got to be careful. Don't use the observation to justify. So you cannot say this that all existing organisms perform glycolysis. That's why glycolysis was in the common ancestor of all living things. That is not a justification. That is basically what I just did. I just rewrite the observation and then I connect it to the justified claim. Yeah. So again, you cannot say all existing organism perform glycolysis. That's why glycolysis was first occurred in the common ancestor of all living things. That is not a justification. Yeah. Okay. So that would be just regurgitating what you're told. So how do you write that then? Okay. So I would talk about adaptation, right? If it's there, glycolysis must have some adaptive selective advantage. Organisms. So you can talk about that. Okay. So for example, you can say that glycolysis glycolysis first occurred in living common ancestor. Okay, and then glycolysis provided adaptive advantage since the common ancestor. Okay, and remained as a very useful trait, right? So for all the organisms that we have today, they still retain the ability to do glycolysis. So you can say something like that. For the anaerobic condition, for the second one, you would have to address that the original Earth was formed under anaerobic conditions. Okay, so so the glycolysis, of course, occurred in the common ancestor of all living things. It must be under anaerobic as well. Okay, there is no oxygen for glycolysis to utilize, right? And then same thing with glycolysis only occurs in the cytosol because the original cell, which is most likely the common ancestor, must have been a prokaryotic cell. Cytosol is the only thing that they have. There is no organelle. Membrane-bound organelles have not evolved yet. So if we look at this scoring guide, let's see how they break it down. Okay, again, it's about twisting logic and words. So let's look at the first one. How do I justify glycolysis first occurred in the common ancestor? So it says, okay, so it says here. So this is basically what we were trying to say. This is pretty concise. It says glycolysis provided a selective advantage that was passed on to the descendants. Yeah. So that's the reason, that's the justification that why all the organisms today perform glycolysis. It's selectively advantage. So it's a trait that was very adaptive that was selected for. Organisms with that trait were selected for, not against. So that's why the trait is now in all living organisms. Second one, anaerobic conditions, right? So you want to say the origin of glycolysis predates, so comes before free atmospheric oxygen. Yeah, so the original condition of the Earth was anaerobic when the first cells were around. And the last one, glycolysis occurs only in the cytoplasm. So same thing, right? So original glycolysis evolved before the membrane-bound organelles. So predates the cell type with membrane-bound organelles or eukaryotic cell, or before endosymbiosis. So you're actually doing grammar and logic here. I don't see there's much understanding of biology here, but you actually have to use the observation and a little bit of background, okay, of what you learned about bio. Remember, the first cell was evolved that was anaerobic, was prokaryotic, Hear me now? Yeah. Hi, Hillary. Can you hear me okay? Maybe? I don't know. Hi, Kyra. Can you hear me?
Yeah, I could hear you.
Okay. Maybe one of the day I should ask all you guys to come in person. I can use the whiteboard. We'll do that one of the day. All right. Sorry. So I don't know what happened or when I lost you guys, but let me continue. Okay. Well, that question we were doing was calculating efficiency for cellular respiration, stuff like that. So let me put this back up again. So was this part C? So this is question number 2, part C. So we're calculating amount of energy available from 30 moles at 7.3 kilocalories per mole times 30 moles. That gives us 219. So there, yeah. So that would be this part here, 219, yeah. And the second part, we should be getting 686, but we only got 219. So the efficiency is about 32%. So I did 219 out of 686. Then the last part is about describing what happened to the excess energy. So as you know, like internal combustion cars, gasolines, is about 20% efficient. Yeah, I used to have an LR4 that can drive 11 miles per gallon. I want to kill myself. Yeah, as soon as I drive out the gas station, I can see the gauge dropping. Yeah, I have to go to the gas station 2 or 3 times a week. That's very inefficient. Now, cell respiration is about 30-40%. 40% efficiency is actually quite efficient in biological system. So what happened is that during energy transformation, every single energy transformation, energy is lost, must be. You have to lose energy, majority of it, yeah? Energy is lost as heat. Okay? If you just stop here, it will be perfectly fine to increase the entropy, yeah? The entropy. That would be fine too if you want to include that term entropy. It's the same thing. All right, so that's the answer for part C. Is that okay? All right. All right, let's look at the next part. So you can see that part C, do you need to memorize anything? No, you don't have to memorize it. Yeah. So 1 point for each bullet. All right. Part D. The enzymes in Krebs cycle function in the cytosol of bacteria. Okay, so this is probably endosymbiosis because bacteria have no mitochondria. Remember, there used to be free-living bacteria, mitochondria are, right? Mitochondria came from a free-living aerobic bacteria that do cellular respiration. It was eaten. Instead of being broken down for food, it became the mitochondria. Same thing with chloroplasts. Chloroplasts used to be a free-living bacteria that was carrying out photosynthesis until it was eaten and became an organelle. So that was the theory of endosymbiosis. So that's what they're targeting here, right? So it says bacteria is in the cytosol, of course, because bacteria only have cytosol, right? So if they're able to do cell respiration, aerobic bacteria, all the apparatus must be on in the cytosol. And any membrane they need will be on the plasma membrane, cell membrane. They don't have electron transport chain occurring in the inner membrane of mitochondria because there isn't a mitochondria in a bacteria. It will have to be on the cell membrane for electron transport chain. But among eukaryotic cells, the enzyme functions mostly in mitochondria. Those are scientific questions. They want to come up with a question that connects the subcellular location of the enzymes in the Krebs cycle to the evolution of eukaryotic cell. So the question is endosymbiosis, yeah? So let's see how you draft this question, guys. So again, you know this is related to endosymbiosis. You know that aerobic bacteria became, you know, so how do you use what's given to write up a question? And the question have to connect 2 things, right? So the question have to connect 2 things: the subcellular location of the enzyme, which is in the cytosol, in the bacteria, to the evolution of eukaryotic cell. Okay, so that's what we're asked to do. So let's see what we can do here. Okay, let me come back to this. Okay, right here. All right, so this is part D. Okay, so how do we put that into words, guys? Okay, so you want to try sometimes if you're not sure what to do, just start trying them. Write something and then build up a simple sentence into a more complex one. So for instance, here, I know that I need to come up with a question. So, are present day mitochondria free living? They use bacteria, so I'm going to use bacteria. Okay. Long time ago. Long time ago. I don't like that because I see the evolution of eukaryotes, right? So our present day mitochondria, free living bacteria, before the evolution of eukaryote. You see that when I say long time ago, it wasn't clear to me. I want to be more specific, so I came back and say, okay, before the evolution of eukaryotes, right? So that would be a question. So I ended with a question mark. So that would be a question. There's no right or wrong question. Okay. So I think that would be okay. Yeah. So that's I connected to the evolution. So our present day mitochondria, once free living bacteria before evolution, and gave rise to gave rise to gave rise to eukaryotic cell. Yeah. Eukaryotic cells. So maybe I add one more, right? So there's probably more concise way to say this, but okay. So let's see what the answer key will score points for. Oh yeah, so here, this is the calculation. We got 219 earlier. We got 31.9 or 32%. You see, they give you anything between 31, 32. So don't worry about sig fig. This is not chemistry or physics or math. Yeah, actually, math you don't worry about sig fig. So this one, and then here's the question. A valid question related to eukaryotic cell. So what is the scoring guy? Okay, right here. So example, since Krebs cycle occurs in the cytoplasm of mitochondria, does it suggest that mitochondria were once prokaryotic cell? Okay. Yeah, you know, I'm reading my answer again. I did not connect the subcellular location. Yeah. So, but it's implied that the cellular location is given. So I'm thinking I'm okay with this one, yeah? Because I'm thinking, do I need to talk about the location of the enzyme? Because that's given. So it feels like if I write the location of the enzyme, the enzymes are found in the cytosol of the bacteria. Does that suggest, okay, so if enzyme, so maybe I'm gonna change it a little bit. If the enzymes cycle are found in cytosol or cytoplasm. Present day mitochondria, free living bacteria before the evolution of eukaryotes and gave rise to a eukaryotic cell. So I don't think we need to do that, but any question, yeah, that at least talks about that kind of endosymbiosis, prokaryotes gave rise to eukaryotic cell will be fine. All right, so that was the most difficult one. So now the new test will ask you to graph on one of those two, yeah, question 1 or question 2. So this is the old test. In my opinion, question number 2 is not valid for the new one. It should be a graphing question, either 1 or 2. So anyways, so you have to graph something, okay? So be prepared to graph. Now let's look at question number 3. Looks like it's an evolution question. You can spot an evolution question right away because seems like you're given this 5 different species and it is the differences in the amino acids. And this amino acid is the cytochrome C. If you forgot what cytochrome C's are, they are used in the electron transport chain for everybody, right? So they are used to transfer electron, part of the electron transport chain apparatus that harvests energy from electrons to do chemiosmosis. So by looking at this molecular structure, by looking at the differences in amino acid, the more different, the more distantly related the organism are. So remember, evolution is a gift. If you get evolution questions, you should always get it correct. So here, you can see the 5 species. Do you see that they all differ So can you tell me which species are most closely related to each other? So if I give you a side question, yeah? So this is the binomial nomenclature. So the first is the genus, the second is the species. So I can see by cheating, these are the same genus. And the data shows that they're only one amino acid different. So yeah, they're the closest, right, to each other. Okay, so they have the same genus. Anything else that's the same genus? No, right? They all have different genus except these two. Yeah. Okay, so that would be the most closely related based on the binomial nomenclature. And don't forget, binomial nomenclature is based on the way they look, organisms' morphology. But if the DNA or protein sequences suggest otherwise, you got to follow the DNA, not by the way they look. Yeah. But lineage binomial nomenclature is based on looks, okay, and structure. Alrighty. Anyway, so this is the first thing popped to mind. So of course, you got to do a cladogram or phylogenetic tree. Now, what is the difference between phylogenetic tree and a cladogram? They're basically the same thing. It's just that cladogram, you have to insert derived characteristics. So for instance, if I were to just do a very similar, very quick one here, okay, whoops, yikes. All right, so we should know some basic classification, okay? So let's say I'm gonna do plants because I always do animals. I've done animals, let's do a plant one, yeah? So let's say I have phylogenetic tree like this. The most primitive plant, guys, is actually moss. Moss, they form a carpet on the bottom of the ocean. They're barely made a plant. They have no vascular tissues whatsoever. They have no roots. There's no stem. There's no leaves. Yeah. And then the next most advanced plant is the fern. Okay. And then we have the gymnosperm. So these are some of the phylums we see in plants. Yeah. Plant kingdom. And then the most advanced and diverse of all the plants are the angiosperms are flowering plants. Okay, so this would be a phylogenetic tree. But if I put in derived characteristics, I will turn this into a cladogram. So if I were to say, okay, right here, vascular tissues evolved. So now you have what? What are vascular tissues? They are roots, stems, and leaves. These evolve, right? So these are organs actually, yeah? So vascular tissues evolved, gave rise to these different plant organs. And then what about put here? What is the difference between fern and gymnosperm? Ferns have no seeds. They use spores. They sporulate. That's why you look at the underside of a fern, you'll see little clusters of spores that can burst and release those spores. That's how they germinate. So seed evolved, another organ, very important organ because the ability for plants now to germinate only when conditions are favorable. That is the purpose of a seed. You have a baby right there, but it will not germinate if it's too dry, if there's not enough sunlight. It will germinate when there's enough condition, the humidity in the soil. So to ensure its survival. So this is a very adaptive evolutionary characteristics. So I'm doing a cladogram, and what is the difference between gymnosperm and angiosperm? Would be flower and fruit, right? You can put either one. Fruit came from flowers, right? So flowers and fruit. So that would be an evolution advantage for a plant to have fruit, because by bearing fruit, you entice the animals to eat you to help you to disperse your seed. Otherwise, all your seeds falls on the same location as you. They will compete with you, right? They will self-compete. That's not a good thing. So the only way is to disperse them either using wind, using water to disperse seed, right? Like dandelions using wind, or using fruit so animals can carry them to other places. So I'm putting together a phylogenetic tree in blue. When I start adding the characteristic, this becomes a cladogram. So that would be an example using plant. Okay, so what do we do now? So we do for this one, right? So this is super easy. You just look at, we already know that these are the closest related. And usually when you do this, you want to identify the outlier, the guy that is most distantly related to everybody else. So if you look at this one, who is the most distantly related, right, to everybody else? So you can see that, okay, so the way they set it up, you can use letters, E, D, so this is EF, right? And this is EA, and these are DGA. I'm just using the first letter. So look like a D is 18, 17, 20, this guy here. From D from G is 18, D from A is this, D from this, right? And if you look at G, G is 3 and 10. If you look at A, is about 12. So your best guess is D. D is most distantly related right here because it's 18 from this and that, huh? And EA and EF is very closely related, right? So it's, your best guess is D. And then two numbers pop to mind. The first is 1, so EA and EF are very closely related, and also 3 pop to mind because 3 is also very close. So they're saying that somehow G and A probably belong to the same clade. Same clade means that they will branch off similarly, yeah? So that popped to mind. So if you draw the phylogenetic tree, you'll see the answer key, okay? That it's you just draw them whatever way you want. You can draw what I'm doing here, or you can draw a little bit differently. Doesn't matter how you draw them, okay? As long as you show that evolutionary relationship. Okay, so I guess I don't have a tree here. I thought the scoring guy would give us a tree. Yeah. Okay. No tree, then we draw our own. Yeah. Oh yeah, there is a tree. What the heck? So you can draw like this. Okay, phylogenetic tree also. But the key is your EA and EF. Remember, they're 1 amino acid different. Remember, G and A, they're 3 amino acid different. So it doesn't matter if you put G on the right or G on the left. Doesn't matter. And D is the outlier. Okay. So when you see very close together, they have to belong to the same clade. Again, the concept of clade is the ancestor, the common ancestor, and all the descendants, every one of them from that common ancestor. That forms a clade. Because EA and EF is 1 amino acid different, they form a clade. Because G and A is 3 amino acid different, they will form a clade as well. And D will be the furthest apart. And how do I know to put G and A first or E first? And you want to see by looking at D. Okay, so if you look at our data again, okay, and if you look at the data, overall, D is further away from GA. Sorry, let me say that again. D is further away from E than from GA. Okay, so if you see that data, if you haven't caught it, I'll be right here. So you can see here, overall, D, this outlier, is farther away from E than they are from GA, right? So about 17 and 18 from GA, but it's 20 from the E guys, yeah. All right. So that's the and you can look at this too. See that? Okay. So you want to put the E, A, and EF farther away. So that would be the right answer. So it's pretty easy to come up with. And you see that you only have 1 point on this. So don't stress out too much. Did I draw this one correctly? The absolute distance don't matter. So all these distances don't matter. Okay. So this is not a cladogram because I don't have derived characteristics. Yeah. Okay. So the next one, what we have to do is to provide reasoning for the placement. So basically what we said, you want to say D is the furthest away from everybody else. So it's an outlier. And you want to mention DG are 3 amino acid different. Sorry, DG. EA are 3 amino acid different. So they form a clade or the closest related. And then EA and EF are 1 amino acid different. So they also form a clade. And collectively, EA, EF are more different than A from E. Yeah, hard to say. But let's see how much you get points for. So I pointed out all the logic, but you don't probably have to do all the logic to get a point. Look what they have. You get 1 point. Okay, for this, for saying that D has the most difference. Yeah, that's it. Okay, it's not a very good reasoning, but you get a point nonetheless. So D has the most difference, okay, in amino acid. Okay, they didn't even finish the sentence. Yeah, from everyone else. Yeah, all right, that's it. Super easy, right? 1 point. So don't stress out about evolution. Hopefully you get one. And then part B, identify whether morphological data, by the way they look, morphology, or amino acid sequence data. So you have two sets of data. Which one are more likely to accurately represent the true evolutionary history by reasoning? So are you on team morphology or are you on team amino acid? Now, if they say DNA, I would definitely go for DNA because DNA is the most important sequence. So there's no right or answer. I believe that you can argue for both. So if you're on team morphology, you have to argue why morphological is better. Okay, so you can trash the other team, right? So if you think morphological data is better, then trash the other one. What's wrong with amino acid, right? So, and if you're on team amino acid, then you trash morphology. You say every organism looks the same. How do you tell from the way they look, right? So that's it. So you just have to identify one, and that's your opinion, and then provide some reasoning to support your answer. There's no right or wrong answer here. You can argue. You are able to argue for either side. So the worst thing to do is to leave it blank. Okay, so let's think about why people use fossil, right? Morphological evidence is the most direct evidence because you can see it, right? They're supported by fossil records, right? Because your fossil record doesn't really have amino acids, right? So you can say that is the reason why morphological is better. You can also trash the other team. You can say amino acids. Well, the amino acids, a lot of different organisms from different species use very similar amino acids, right? Like horses use hemoglobin. A rat uses hemoglobin. A human uses, how do you tell, right? How much of difference in amino acid differentiate the species apart? Maybe that's not as good, right? Okay, so anyway, so let's see what they say, yeah? So you got, and provide reasoning. So 2 points here. So let's say identification, amino acid. So you can say that, so you trash morphology. You say morphological, by the way they look, may be similar, right? Okay, even when there are big differences in amino acids. Okay, do you have to say why they're similar? You know, that would be great. So you can say convergent because they live in similar environment, they converge, they become analogous structures. They look similar, but they actually have different ancestry. So that's a great one to include. But if you forgot to include that one, it's okay, right? Okay, and you can also say that if you are for the other team, so if you support morphology, so you can say, I support morphology, better data, right? Then you can say that similar molecular sequences may result in different molecularity or an example of species with similar proteins but have different forms, yeah? So you can say chimpanzee and humans have very similar proteins, but obviously they're different structure, they're different species, right? So that means you trash the amino acid data. So just simply put that, okay? You don't have to come up with a perfect answer. As long as you can support your reasoning that is somehow not fantasy land reasoning, then you're good, yeah? So, all right, so that will be it, okay? So if you can, guys, if you did the test, you should add up how many points you have, right? Okay, so I'm gonna start collecting data, yeah? And make sure you're able to write these on your own. Let's look at the next one. Question number 4. Question number 4, both mitosis, meiosis are forms of cell division that produce daughter cells containing genetic information. Okay, sorry here, let me see what it says. Containing genetic information from parents. Great. Describe 2 events that are common to both that ensures the resulting daughter cells inherit the appropriate number of chromosomes. So you're going to describe 2 events that's common, but a little bit more detail. What about the 2 events? To ensure that the daughter cell inherits appropriate number of chromosomes. So you can basically say anything here. What is common between mitosis and meiosis? For example, that DNA synthesis, DNA replication occurs prior to them. Okay, but you know, this is not exactly happening during mitosis, meiosis, but it is an event that is common to them that they have to replicate the DNA so that the daughter cell gets the right number, right? Or you can say that anaphase occurs. right? So anaphase occur to separate the chromosome so that mitosis get identical chromosomes, and anaphase happen twice for meiosis so that the daughter cell have the right number of chromosomes. So you can talk about anaphase. You can even talk about prophase is common, right? Prophase sends out spindle fibers to make sure that the spindles are properly attached to the chromosomes, right, to the centromeres, so that they can be lined up properly so that the separation is appropriate, right? So basically, you can talk about any stages that you see fit, as long as you can connect the dots. That's what you need to do. Okay. If you want to talk about prophase, connect the dots. What does prophase have in common to make sure that they have the same number of the right number of chromosomes, right? So let's look at the answer key says, what the answer key says. So here, you get points for talking about the spindle fibers. They form and they're attached to chromosome, right? They just want you to describe it. You don't have to justify. If I were to justify this, then I have to talk about that the chromosome must be moved and lined up at the metaphase so that they're properly separated without error. That would be a justification, but the description, you don't have to go that far. Okay, you can even say chromatin condenses into chromosome. Remember, chromatin is loose, diffused form of DNA. They have to condense to form chromosome. You can even talk about lining them up in metaphase. You can talk about the separation of the chromosomes. You can talk about before. So you can talk about G2M checkpoints. So you got to do the cell cycle checkpoints to make sure nothing goes wrong, so that the daughter cells have appropriate number of chromosomes. And the first thing that came to my mind was the DNA synthesis. So that is okay. So they come before both mitosis and meiosis. That is an event to ensure that they get the appropriate number of chromosomes in the daughter cells. You can even talk about cytokinesis. So you can talk about anything. Yeah, okay. So just put something down, you get points. Next one, part B. So this is a 4-point question, okay? So describe 2 events, so 1 point for each event. And part B, the genetic composition of daughter cell produced by mitosis differs, right? They're identical to each other, but not in meiosis, right? Describe 2 features of cell division that lead to those differences. So this is something that I talk about every week. The source of genetic variation is mutation, crossing over in prophase 1, independent assortment in anaphase 1, and random fertilization. So the 2 features would be crossing over between homologous chromosomes that shuffles the parents' genomes, right? So that's why meiosis is different. The genetic, that's why meiosis produce different gametes, right? You can also talk about independent assortment, right? So those 2. You can talk about more. I think there's more features that you can talk about. You can talk about synapse, crossing over. Let me see, differ from that. And you can talk about mitosis does not have them. So let's see what the points are. So these are easy points here. Okay. So yeah, you can talk about 1 division, 2 division. Again, the differences between mitosis and meiosis, right? So mitosis at 1 division, you get 2 cells, whereas meiosis, you get 2 division, you get 4 haploid cells. Okay. So you can talk about chromatids separate twice, okay, in meiosis. Sorry, I shouldn't say that. What I mean is that, okay, crossing over does not occur in mitosis, but do occur in meiosis, right? And homologous chromosomes separate, that's independent assortment. They don't in mitosis, they do in meiosis, right? So that is the 2 most obvious one. You can talk about the number of cell division or number of cytokinesis. So those are easy points. Stay away from the chromatids, because chromatids are not chromosomes. And when chromatids separate, they become individual chromosomes. So stay away from that unless you understand what chromatids are. So do the obvious one. You just need 2 to talk about, okay? So it's very easy, right? So those are all what I call 3 points. Okay, let's do the next one. Next one is an experiment. Look at the setup here. So I do want to talk a little bit more about this. This is about plant hormones, yeah? So plants do produce hormones, and that also affect their physiology. The same way hormones affect animals' physiology. So plants have hormones, plants have immune system, just like animals have immune system. Plants do not have nervous system, yeah? Even the Venus flytrap that they move to engulf a bug that falls within it, that is not nervous system, okay? That is simply change in osmotic pressure that causes the leaves to close. Okay, so that is not a nervous system. So plants have immune system, plants use endocrine system, plants do not have nervous system. So this question is about one of the experiments. It's actually quite famous experiment. It's conducted by Charles Darwin, the guy who come up with evolution. Yeah, he also played with a bunch of plants and experimented with them. Other than bird watching, Charles Darwin did this experiment. So here is a process called phototropism. If you look at the sunflower will follow sunlight throughout the 24 hours. It's able to bend towards light to maximize photosynthesis. So that's the phototropism. It's the behavior response that plants have. Okay, so don't use phototropism for animals. When an animal swims towards light because there's where food is, that is not phototropism. Okay, that is called kinesis or chemotaxis. So let me just talk about those two words so that we don't get confused with them. So you can talk about phototaxis. That is animal behavior. So this is the most photo. I forgot some letters here. So phototaxis, chemo, moving towards chemical, bacteria. Okay, moving towards chemicals. Yeah. Let me plug in my laptop. Sorry. So here you go. All right. So phototaxis, chemotaxis, if you have a bacteria moving towards a favorable food chemical or moving away from a toxin. So this is how animals do it. Plants have tropism. So if you see this word tropism is plant response. You can have phototropism, moving towards light. You can have hydrotropism, moving towards water, right? So roots grows towards water, and gravitropism, towards gravity. You see that roots exhibit positive gravitropism. Stem have negative gravitropism. So positive gravitropism means you go towards gravity. Gravitropism. And negative gravitropism is like stem. Stem goes against gravity. Gravity goes down, stem goes up. Okay? Even if you tilt a plant sideways, stem still grows upwards against gravity. It's not going to grow sideways. Yeah, that's not how plants behave. So they have negative against negative gravitropism. And one more is like vines, grapevines. They wrap around whatever they touch. That is called stigma, which means by touch, tropism. Yeah, that is a plant with very quick response. Okay, to if you look at the Venus flytrap, if you look at the documentary, when it opens up, there's little tiny hairs. So if the hairs are touched twice by insects or bugs, they will shut off on its own. Yeah, that is a response used changing the osmotic pressure. So it's not a nervous system, but it's a tropism. Yeah, so plants do those. So tropism, and animals, bacteria, they do taxis. Okay, so don't confuse those words. So let's look at this, phototropism, bending. So phototropism is a response in which plant shoot, which is the stem, grows towards the light source. The results of 5 different experimental treatments from a classical investigation of phototropism are shown. So let's look at the experiment, guys. If you cut the tip off, there's no more phototropism, right? So do you see 1 will be the control for 2? 1 is the control for 2. 2 is the experiment. The experiment is the tip necessary for phototropic behavior. If you cut the tip off, there's no more bending. So you need the tip, okay? And 3 says the tip is covered by opaque cap. So that means opaque means kind of you can't see through, right? Not clear, not transparent. So you see that the tip responsible, you have to be able to detect the light, to see the light, quote unquote, right? Quote unquote. So you can't really, plants don't really see with the eyes, but they have to be able to detect light. So if you cover it up, you don't chop it off, the tip is intact, right? Unlike experiment 2, you chop the tip off. Experiment 3, the tip is intact. You just covered it up. So if the tip cannot see light, they will not bend. Let's look at 4 and 5. That's a little weird. So 4 says the tip is separated and connected by a permeable barrier and then 5 is an impermeable barrier. So 4 and 5 kind of contrast each other. So if you have a permeable barrier, then it will bend. So something from the tip must be able to communicate with the rest of the plant. Okay, because permeable means things can go in and out. If you block it, if something at the tip is stuck at the tip, or something from the bottom of the plant is stuck at the bottom of the plant, unable to go through, then there's no phototropism. Okay, so that's what the experiment is. So it turns out, guys, it's actually a hormone produced at the tip that travels down. The hormone is called auxin. You don't need to know the hormone to answer the question, but auxin is one of the most important plant hormone. It's responsible for phototropism. It is made in the tip, synthesized at the tip of the plant, and then it has to be able to travel down. Okay. And then what this does is that it causes the dark side to elongate. So for example, this is the side away from the plant. Imagine the dark side elongate. The cell becomes longer by gathering more water in the central vacuole. So the dark side elongates, so that's how it bends, and bending occurs. So it's not cell division, it's cell elongation. We're not dividing more and more cells on the dark side, but just by elongating them, you can bend the opposite direction. Yeah. So when the right side elongates, you'll bend towards the left side. That's how plants do phototropism. It's this hormone. Yeah. But we don't need to know this hormone to answer the question. Remember, it's not about memorization, right? The whole entire AP class. So let's take a look at this. Number 5A, support for the claim that cells located at the tip detect light by comparing the results from 1 with the results from 2 and 3. So what is 1? It's a control. For 2 and for 3. So 1 and 2 shows that the presence, the physical presence of the tip is necessary. But 3 does not show the physical presence. 3 shows that the tip must be exposed to light in order for, and without that, there's no phototropism. So there is a difference between 2 and 3. Again, you're asked to compare, but the tip detect light, right? So 2 shows you that the physical presence of the tip is necessary for phototropism. 3 shows you not only that, but the tip must also be able to sense the light. It has to be exposed to light. Otherwise, phototropism does not occur. So that's the difference between 2 and 3. That's why the opaque cap, which is blocking the sunlight, even when the tip is physically present, if you block the sunlight, the tip is unable to sense or see the sunlight. There is no phototropic response. So that's basically part A. 2 points for putting that together. So basically interpret 2 versus 1 and 3 versus 1. Okay. And if you look at part, let's look at the answer key, what they say. So again, when we put that into words, now it will be a little bit more difficult. So I would say, so here, support in treatment 2, tip is removed and plant no longer bend towards light. So tip is necessary, right? And then in 3, the cap blocks the light to the tip, and the plant no longer bend towards light. So it's unable to detect light. So light is necessary. Light detection is necessary for phototropism. So it feels like they didn't really say much, but that is basically enough. Okay, I would elaborate a little bit more. Okay, let's look at the next one. That's 2 points there, guys. Next one, in treatment groups 4 and 5, the tip of the plants are removed and placed back onto the shoot, so like crafted back. There are actually people who do this to trees. Yeah, they cut the tree off and craft on some other trees. So you get the plant, you get a tree with multiple fruits. Okay, plants can do that. We cannot. That's called crafting. Okay, so we have 2 layers. Again, you have the permeable one, so nutrient can go, chemical can go up and down the tip, and the other one is impermeable when you cannot. Okay, so the question is describe 2 additional characteristics. So what are the 2 additional characteristics of phototropic response? Okay, so from 4 and 5, one is that chemicals must be able to diffuse, right? Throughout the plant or from the tip to the rest of the plant in order to see phototropism. Yeah. Okay. So let's see what the answer says here. Yeah. So right here, that was my second point. The substance must diffuse from the tip to the rest of the plant in order for plant to bend to exhibit phototropism. Okay. And the last one here, the second one, you need to give two additional one, right? The tip produces a substance in response to light, causing the plant to bend. So they're saying that, okay, it's not the rest of the plant, it's the tip. Because if you look at the rest of the plant, when they're not joined to the tip, they don't have phototropism. They're unable to bend. So there must be something produced at the tip. If you compare 5 and 1, when the tip, if you look at 1, 2, and 5, it shows you that there's something at the tip that is required. Okay, you look at 1, 2, and 5. Okay, if you cut the tip off, it doesn't bend. If you don't let stuff from the tip to pass through the rest of the plant, it does not bend. So 5, 2, and 1 shows that there's something produced at the tip that is required for phototropism. So this is a little bit harder because the two additional characteristics, sometimes we're not sure what it is. Even if you only were able to write down one, you still have most of the points here. Yeah. Okay, so that would be question number 5. Let's move on. Okay, let's look at question number 6. Okay. So question number 6, let's look at this. So we have, we want to rescue small isolated population of snakes from decline. And any small isolated populations are subject to genetic drift. Okay, or any of the disaster can wipe them out very easily. Yeah, changing environment, for example, natural disasters, for example. A few male snakes from several large populations of the same species were introduced in 1992. So let me look at the graph. So 1992 is this arrow. So you start to introduce male snake. Yeah, and do you see that I have more, and after the introduction, seems like 93, 95, 97, 99, the population start to rebound, okay? So it is doing something, right? So again, you introduce a few male snakes from several large population introduced, the snake reproduced sexually, and there were abundant resources. So food is not a factor. So you cannot say that they didn't grow because there's not enough food. When it says there's abundance of resources in the environment, you can't say that. That's not the reason for the decline of the original population. It must be because of genetic drift or some other reason, yeah? So the figure shows the study. Okay, so let's see what we want to do here. So one is describe one characteristics of the original population that may have led to the population decline. So once again, you cannot talk about not enough food, okay? But other characteristics that can cause this to decline. So I gave you one, like genetic drift. Let's see what the answer key says. Okay. So lack of genetic variation is possible too, right? Because small, oh yeah, right here. So the first one I actually mentioned lack of genetic variation. So not genetic diverse enough, yeah? And then, okay, I talked about genetic drift, but I didn't say what is caused by. So when I was thinking about genetic drift, I was thinking about that in terms of the lack of genetic variation. Yeah, because a small population without enough variations, that could cause them to potentially inbreeding. Okay, and when environment changes, then there's a problem. You can also talk about this. Was an aged population. There's not enough young snakes. So how do we know that? By reading the question, guys. If you look at the question here, it says a few male snakes, right? From larger population introduced. And then if you look at the juvenile snake captured, is also high. Total snake captured is high. So they're saying that there's not enough reproduction. Here, the figure shows the result of the study before and after introduction of outside male. So you added more male. So, and then population bound. So male snake was the key. Yeah. So male snake was the key. You added the male snake, not female snake, and then population rebound. So there was not enough male reproducing. Okay. So had unfavorable sex ratio, too few males. That was the big clue. Okay. And then you can even be as this one, possess a harmful mutation or disease. That is not seen in the question, but it is a possible reason or cause for the population declining in the original. So declining something killing it, right? You're not in a Hardy-Weinberg equilibrium. There's a disease, yeah, on the snake. So you want to think about using common sense, but it has to be one that makes sense. So I think the best answer here is probably too few males, because the addition of the male snake is directly what causes the rebound. You cannot talk about not enough food when it says food is plenty. Now, part B, propose one reason that introduction of outside male rescued the snake from decline. Well, that increases genetic diversity, right? That caused more mating, more reproductive success, right? And that's the answer. Yeah. If you talk about the mutational disease, then maybe the male brought in a resistance gene. So now when they are, so you have a migration of gene flow, right? Gene flow moved in, which carry resistance gene. Now that is now brought to this declining population from the disease. If you want to talk about disease as a reason, okay. So that would be another point. Okay. All right. Let's see the last one. So these are 3 points questions. On the new AP, there's no more 3 points. There's always 4. And finally, how does the data support the statement that there are abundant resources in the environment? So how do we know? How does the data is the graph. How do I know from the graph that there's plenty of resources? Because I see an exponential growth. Yeah? The problem is not the resources. As soon as the males are introduced, several years later, the population is growing exponentially. That's why it looks like the S curve, right? So that suggests that resource is not an issue. They are able to grow when possible. So that would be the first thing popped to mind. Let's see what the answer key says. So it says population can and does grow since the introduction of the snake, right? If the resources are limited, then the population could not have grown in the first place. So that would be a good answer too. All right, so 1 point. So you're looking at the data, interpreting based on what you know, yeah, based on what's given. So you're not really learning about. You don't have to read the whole chapter about biology, ecology to do this, yeah. So common sense. Let's look at number 7. It's about smell reception, yeah. So smell is the olfactory bulb, is linked to our memory, limbic system, yeah. So memory and smell go together. So smell reception in mammal involve interaction of airborne odorant molecule from the environment. All right. So the receptor proteins detect the chemical and then trigger sensory neuron to interneurons in the brain to motor neurons. So you can do whatever you want. Yeah. Okay. The binding of odor molecule to the receptor triggers action potential in the olfactory neuron, sensory neurons. Results in the transmission of this information to the brain for interpretation. What am I smelling? And it says mammals, their genomes typically have 1,000 functional odorant receptor genes. So you have 1,000 genes dedicated to the sense of smell, right? Some mammals, I'm sure dogs, have a lot more than, say, humans. Each coding a unique odorant receptor. So you have at least 1,000 receptors. So part A, so all these information, guys, they're clues. Give it to you, background information for you to process information. So don't just skip all these questions, yeah? All these reading. So describe how the signal is transmitted across synapse from an activated olfactory sensory neuron to the interneuron that transmit information. So we talked about this in the nervous system. Across the synapse is the physical space between 2 nerve cells. You have to use neurotransmitters, right? So the neurotransmitters are released into the synapse, which will bind to the chemical, give you the light receptors, those neuron or cell. That's basically it. That would be pretty detailed already. Do you have to say calcium rushes in, neurotransmitter feels vesicle fuse with the and release the neurotransmitter into the synapse, and synapse diffuse across the synapse and bind to the voltage gated, ligand gated channels, that would be a little bit too detailed, I think. Yeah, this is just for one point. So again, neurotransmitters released into the synapse is able to carry and transmit the information to the next neuron. That would be enough. Okay, so no fancy details. Yeah, you don't even need to name the neurotransmitters. Yeah, you don't have to know some of the neurotransmitters are excitatory, some are inhibitory. You don't need to do that. So let's see what the points are for. Okay, neurotransmission across the synapse. Okay, so it says here neurotransmitters are released from the olfactory neuron, sensory neuron, and bind to the receptors in the postsynaptic interneuron. That's what we're told. Yeah, that's good enough. The key is the neurotransmitter release into the synapse. All right, let's look at second part. So second part, 2 points. See the 2 bolded letters. Explain how the expression of a limited number of genes, odorant receptor genes, can lead to the perception of thousands of odors. How you have a limited number of genes? I guess 1,000 is not a lot. Compared to all the chemicals out there you can smell, 1,000 is not a lot. So how can a few genes, 1,000, allow you to perceive thousands more odors, right? Use the evidence about the number of odorant receptors genes. So they want you to use the evidence about this 1,000. Yeah. What do you guys think? How can you have limited genes but able to detect so many different various chemical odorants? The first thing popped to mind is alternative splicing, right? Remember you have the same DNA, you transcribe that into the primary RNA, but you do alternative splicing by splicing the different exons together. So the same DNA can produce different type of receptors. So even though there's 1,000 genes, you can alternatively splice them, okay, to produce different combination of the exons. Every combination of exon gave rise to unique receptors. Second thing popped to my mind is that your brain, depending on which region of the brain, so it's the combination of the receptor. Let's say you trigger receptor 1, 5, 7. That could produce a different sensation in your brain than if you trigger 4, 5, 6, right? So it's the number that the different number of receptor that's triggered can be integrated by the brain to produce different sensation. That's like a BS, but that's how that actually works, right? If all these receptors, the location of the receptor matters also, right? So let's see what the answer key says, but I will stick with the alternative splicing, yeah? So you have to explain and support. So you can talk about, so here's one I did not talk about. It says one odor molecule can be recognized by more than one receptors. So multiple receptor can multiply one, or one odorant receptor can bind to multiple. Well, that's a good one, right? Because, okay, you have 1,000 receptor because you have 1,000 genes. But what if this odor receptor can bind to multiple odor, right? Multiple chemicals. That means you detect more than 1,000. Yeah? And your support is what? That there's many combinations. Expand beyond the possible order detected given only 1,000 genes. Okay? That's the one I did not talk about. I did talk about the genetic aspect. I did alternative splicing of RNA into different possible combinations of exons, which can produce a lot more protein receptors than the limited 1,000 DNA. Okay, so multiple receptor can be produced from the same DNA, alternative splicing. And I did talk about the central nervous system control. So signals are integrated in the brain. So multiple interaction among neurons in the brain can give you different sensations. Yeah, so that's kind of like the most BS one, but it is true that you, depending how you integrate in the brain, the network can trigger different responses, right? Different sensations. All right, so that's number 7. So let's look at the last one now. Okay, last one is immune system. So individual has lost the ability to activate B cells. So what are the B cells? B cells produces antibodies. So B cells, once they're triggered, become plasma cells. Their job is to produce one type of antibody that's called monoclonal antibodies. So the antibody will circulate in your blood. Like right now, if you're vaccinated, you have a bunch of these antibodies circulating in your blood. When you get exposed to COVID, these antibodies will mount an attack, will bind to the COVID virus themselves and trigger phagocytosis, right? Neutralize the COVID viruses before you feel any symptom. That's the purpose for vaccination. Now, this person does not have this ability. So A says propose one direct consequence of loss of B cell activity. Okay? And the B cell had to do with the humoral. T cell, the cytotoxic killer cell, is cell mediated, the two branches of your specific immunity. Okay, so we're talking about the B cell. Okay, so initial exposure, unable to produce antibody, right? Okay, that would be a very easy one. And propose one direct consequence of loss of the B cell. Second exposure. Okay, so it's not just the first exposure. You're unable to produce memory cells, so you will not have a secondary immune response to it, right? So second exposure, right? So if you look at, and then the last one, describe one characteristic of your immune response that is not affected by the loss of B cells. So you can talk about the T cells are still active. The cytotoxic killer cells are still active. You can still target an immune response using those T cells against viruses infected your own cell that is infected by viruses. Remember, you got to get rid of them because they are the factory for viral reproduction. So your T cells, the cytotoxic killer cells, will kill your own infected cells so they don't reproduce viruses. And your B cells will produce antibodies, and antibodies will circulate in your blood to target directly the virus themselves. So let's just look at the last part for those ones. So they want you to understand how B cells and T cells work. All right, so the initial response without B cell is you do not produce antibodies, you do not produce memory B cells, okay? Second one, what about the second exposure? All right, actually, I should probably look at the question a little bit more closely. The second exposure means that we cannot mount a second response fast enough. Remember, the whole purpose for second exposure is that second exposure is super fast, so you don't feel anything. So you're unable to do that without the B cells. And also, you do not have the kind of response, right? The second exposure, usually the B antibody load is like 100 times greater. And you're unable to do that anymore. That's actually a better answer. I should have thought about this question too, a little more closely, okay? So you can mount a second response at the same speed, okay? And the last one, you still have cell-mediated response. You can even talk about the non-specific components. They're still active, right? So there's a lot of things you can talk about. So basically other components of your immunity. Alrighty. So guys, if you know your scores, so please keep them. Because I don't want to spend time during class adding up your scores right now. I just want to get the questions done. So we're already out of time. So do the next week's test. We're almost going to do a multiple choice. We're doing full response to get used to these kind of question types. And I'll see you guys next week. Okay, guys? Okay, thanks, guys. Bye. Okay. Let's take a look at this. And